Welcome to Data Skeptic Animal Intelligence. When we first started planning this season of the show, obviously it's a lot of brainstorming, and part of that brainstorming was a list of species that I was hoping we could find someone to talk to about. Bees, parrots, dolphins, and octopuses were my top 4, as I recall. Octopuses made the list in particular because of this distributed nervous system they have. The arms can act independently. That's fascinating. I heard almost unbelievable things about how their sensory system worked. I get the lowdown on that and a whole lot more in today's interview.
I'm Tamar Gutnick, and I'm visiting research in the University of Naples in Italy. My main topic of study is octopuses and the octopus nervous system and behavior. And so neuroethology, how the nervous system connects to behavior. I deal more with cognition and learning behaviors.
And I imagine there are many ways one could study these things. Maybe not simulating them in a computer, but laboratory settings in the wild, all these kinds of things. What's the nature of your research?
I do laboratory research. Octopus research in the wild is a very new topic because technology had to catch up to the octopuses and to see creatures in general. I do lab research. With behavioral paradigms, we keep octopuses in the lab, and then we do behavioral paradigms, and we also do some neural recordings.
Well, I've heard stories that octopi will get out of their cages and wander around and things like this. Are there any complications or challenges in your work?
There are many. Many. The moment you put saltwater aquariums into a room, complications begin there. I think one of my favorite anecdotes from working with octopuses with behavior is that we always have to cover our cameras with nylon, with plastic, because the octopuses enjoy spitting on them. And they're very accurate. And they'll also spit in your face. It's something that they use in natural environment. When they want to push something away, they'll spit water on it. But they have learned to do it outside the water. So everything has to be covered in plastic.
So maybe it's dangerous to anthropomorphize, but how do you interpret that? Are they shy? Are they angry? What's the reason for the behavior?
It would be a very, 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 very anthropomorphized answer.
Sure.
They're not all shy, and they don't all do it. Different octopuses behave differently and react to us differently. I think they do it because they get a response. They learn that they get a response. I don't think they understand they can read our response, but it's clear when people start moving around and stressing out. You start moving differently and trying to cover stuff and wiping your face. So I think you can see a response, and that is a nice trigger. But not all octopuses do it. Some animals will do it. Some will be bolder. Some will be very hard to get out of their house. And that's another one of the things that's very interesting about them, because they're not all the same. And you don't get an octopus and you can just reproduce the same thing over and over again.
As you work with them, do you develop an ability to recognize them as individuals?
When you work on long experiments with them, yeah. You know which ones to, which ones are going to steal the tweezers, which ones are going to need the time, to come out, to get them to actually participate in the experiment. And the more you work with them, the more you realize the differences between individuals. But yeah, again, in the lab, they have a limited time in the lab. So part of what we do always when we work with them is also to be the ones that actually feed them and care for them, because that gives you a good idea about the individual animal. And when you put them into an experiment, if you set up behavioral task, having that idea and having that communication, and it's not really communication, but the animal is used to you being around and the way you move and the speed you go in and things like that.
Well, you're recognizing them individually. I suppose what I'm going to ask next is a whole new line of research, but just in a hand-wavy way, do you think they recognize you individually?
That's a tough question. I'm not sure. I mean, there is a recognition of different people. They react differently to different people. And that's clear from just working on them. And all of this stuff, I mean, and a lot of the what we know about octopuses is very anecdotal because there isn't that much research. I mean, everything in a way is new. But if you worked with them for many years, you get an idea and you know sort of what you need to do. And it's clear that they react differently to different people. What it is that they recognize, how they recognize this person is different from someone else, I don't know. I don't know if they recognize an image or they recognize the way people move. That's something I don't know, and that's I guess topic for research. In a way, what is it that they recognize, and why would it be important for them to recognize faces of humans, for instance? But we know that other animals do that.
Yeah.
I mean, that sheep recognize human faces very accurately, for instance, and other animals as well. So whether that has a real ecological reason, it might just be a perceptual ability.
Well, you'd mentioned they spend a limited time in the lab. Could you expand upon why that is?
Well, the fact is that at the moment, there is only one way to get octopuses into the lab, and that's, and I'm talking actually, right now, I'm talking about the European animals. The European animals is Octopus vulgaris. When you work with octopuses in Europe, they are wild caught, and there is no way to telling how old they are. They have a pretty short lifespan. And once they reach reproductive age, that's the end. They enter a stage of senescence, which is a deterioration, age related deterioration. In America, people work with other species that are actually can be bred in a lab. And so you have a better idea of what happens. But in general, they're pretty short lived animals. They're not like my turtles, put it this way.
Well, my next question probably has no precise answer, but I want to ask you anyway. How intelligent are they?
I've been talking about this lately quite a lot, and then in all my discussions, we've come to the conclusion that there really isn't a singular definition of intelligence, right? Mm-hmm. What is intelligent? What's intelligent behavior? What we know about octopuses is that we see behaviors in them that we see in vertebrates, and we don't see, they are an invertebrate. They are completely outside our evolutionary line, and they're outside the evolutionary line of most animals that we consider intelligent. But they have behaviors that we recognize as a sign of intelligence, problem solving, learning quite rapidly, everything to even play behavior. But that doesn't really give us an exact answer to the question. What I also think about is one of the issues about us understanding the intelligence of octopuses is that we don't really understand what their sensory world is. We don't understand how they perceive the world. And that's something that is just developing now with more and more research going into the sensory organs. A lot of how we look at animals is from our perspective. And octopus perspective is so completely different. So I think a lot of what we need to understand is really what their sensory world is. What are they using to base decisions on? And that's something that we're really just finding out more and more about.
Well, it seems like we could assume they have some version of our five senses. Maybe not smell, but probably taste for sure, touch, some sight, some sound. Do they have sixth, seventh, and eighth senses as well?
No, but they have the sense of smell, they have the sense of touch, and they have them completely outside the central nervous system. They smell and touch with 8 arms, and they have this peripheral nervous system that cannot be compared to anything that we know or understand. And they have photoreceptors there too. They have chemical receptors. Whatever goes on there is something that is not comparable to any other animal. And that's what makes them fascinating also, because we can't compare them. So the vision sense is something that we can understand. And a lot of tests have really looked into the psychophysics of vision. in cephalopods in general and octopuses in particular, because it's easier to test. But what goes on in the arms, which is 2/3 of the nervous system, is, I mean, really in the last 5 years, we know more and more receptor types that are being found, whether it's photosensitive, tactile, chemical, all of these things. It's just something that you can't compare. It's like smelling with your finger, or I mean, that's the thing. There is no comparison.
Yeah, I can't imagine it.
There just is no comparison, and so we can't imagine it. And in a way, when you have 8 of those arms with hundreds of suckers and they're moving everywhere, then you don't even know what is it in the sensory stimulus around them, because they're moving everywhere, that catches the animal's attention. Because at some point, your animal, which is moving around and touching a million things, suddenly something is grabbing their attention, and you don't know how that happens. What is it that triggers the fact that R3, actually R3 is not a good one. R3 is the reproductive arm in males. But I mean, why L2 has suddenly captured the octopus's, the central nervous system of the octopus's attention. And it's fascinating, but it's very hard to study, which is why I think it's taken quite a long time.
Yeah, it seems like their nervous system is very exotic compared to other things we would describe as intelligent. In particular, it's like it's not so centralized. It's like a distributed nervous system. Or is that a fair description?
Yeah, I mean, 2/3 of the nervous system is outside the brain. If you look at the vertebrates or anything else we consider intelligent, then we have an idea about the organization. There's a forebrain, there's a midbrain, there's a hindbrain. We have an idea about the organization, and it follows a certain theme or a certain plan. If we want to look at any of these vertebrates and look for a certain sensory input or output, we have an idea where to look. With octopuses, we know that they're solving the same problems as other animals, and the ones that share their environment specifically, like fish, teleost fish, or cartilaginous fish. So they're sharing the same environment, they're solving the same problems, and we know that they have neurons and they have action potential. But all of this middle part is very different, and that's something to figure out.
Well, I'm sure you can observe them with video, but what other forms of measurement can you take?
We work a lot with video, because that's the behavioral aspect of it. My work in particular, we also do neural recordings. So we implant electrodes. We developed a new method for planting electrodes and implanting basically a logger into the animal, and then he can freely behave. And afterwards, we basically download the brain activity. And that's something we started very recently.
Could you expand upon that? Are you able to observe the same specimen for long periods of time, for example?
Well, no. This is it's a terminal experiment, so it needs specific approval, because there's one experiment that you can do with the animal. Basically, what we can do is we can get a logger into the animal, and we get up to 12 hours of neural recordings together with, we synchronize that with video plus motion data from the logger. These were built for homing pigeons originally, because you can't collect the data online while this pigeon is flying from Russia to Switzerland. So these were built for that, because when you have the logger in the animal, there's no access to it. And so we get about 12 hours of recording. Now we're working on improving that. But again, it's very new.
Do you have specific plans for where you'd like it to go?
But going back to the idea of the brain and not being able to have the same idea of what to look for where in the central brain, we do have some ideas. And these are often a lot of this research was from around the 1960s, 1970s, in which time they did lesioning experiments, which are not the most accurate, but we have certain ideas about different places. And so the place where we originally implanted these electrodes is what is considered a visual learning center, an area that's associated with visual learning. And so the idea is to continue in that vein and look for activity during learning or memory recall.
And when you then retrieve the neurologger's data, could you talk a little bit about what the data is? So, like, what are the measurements, I guess?
It's basically it's voltage. We measure the voltage, we have the amplitude, and this is over a 12. I mean, there's a lot of data.
Sure.
It's an immense amount of things for a very, very long period of looking and comparing videos to the electric activity. And then it has basically it's all voltage over time, pretty much.
And do you need to do any pre-processing or analysis on that, different techniques?
Well, I have my expert who does it, who's a lovely biophysicist. But basically, when you take this stuff, the first thing we had to do is because there is nothing to compare it to. So a big part of our work was to just prove that find that this isn't motion, because artifacts are one of the biggest problems in electrical recordings. So whether it's your neon light or muscle movement. So a lot of the work went into excluding artifacts from the data.
Sure.
And what was nice is that basically it's clear to us that what we're gaining is brain activity. At the moment, we don't know what that activity is related to. We have activity, but we don't have a specific behavior that it's related to.
So if it were motion, you've got an easier way to do it. The camera can tell you motion. The implants, you want to measure something deeper or more informative, like the brain activity.
Yeah.
Is that then equivalent to like an EEG or something we might do on the human brain?
It is an EEG. Mm-hmm. It's basically EEG recordings. It's a local field potential. In EEG, a lot of times you have surface electrodes, and these aren't surface electrodes, they're penetrating electrodes, but basically it's recording local field potentials with penetrating electrodes. And that's what they do. I mean, this is the same thing that they record, for instance, in these migrating birds, where they find that, well, it turns out half of it sleeps and the other half isn't sleeping because it has to keep flying. Unfortunately, not that simple in an octopus.
So can you take any inspiration from neurology, or are they too exotic of a species?
They share the building blocks. It's not like they're completely different. They share the building blocks. They have LTP. I mean, sorry, I don't know. That's very jargony. Long-term potentiation, which is a cellular form of learning. So that was shown in brain slices, and they show that if you have stimulus in a certain, like concurrent stimulus or in rapid stimulus, then they show a cellular form of learning. So there are a lot of things that we look at, and we look at the same things. I mean, we look at waveforms, we look at spiking activity. We do know that they have spikes. So we can learn a lot from what we know from other animals, and also we know a lot from other invertebrates. I mean, there's a lot of work done on insects, of course, but also aplysia. Most of what we know from about the basics of neuronal transduction was done on giant axons from squid, right? So all of that, that's something that we learned on a cephalopod. The same for sensitization, for instance, were experiments that were done on aplysia, which is a relative, slightly less spectacular, sluggish relative. So there's a lot to learn, and there's a lot to learn from very old experiments because people did a lot of work. It's just that the technology was not there. The technology was not there, and it wasn't there not for all animals, but for octopuses, because octopuses are unique in the fact that they have these 8 arms and they have no skeleton. They have nothing to anchor anything to, and if it's not anchored, they'll just pull it off in seconds. And you really want to associate behavior with neural activity. And for associating behavior with neural activity, you need the animal to behave.
And I'm wondering if you would frame sort of your perception of, I guess, the quality of the data or the precision of it. I was once shown an MRI machine that, well, 2 scans. One was 5 Teslas, I think, and one was 8. And side by side, 5 looked really blurry. There was so much richness of data. Where are you at on the journey?
In my case, we are very early on. We're very, very early on because the technical issues are many, from the type of electrodes that might be possible to fix properly, to recording speeds that depend on your loggers, which now we have a slightly better or more advanced version of, to the fact that it is not very easy synchronizing video with a logger that is inside the animal, inside salt water. Most frequency signals are just not going to go through it. So we're very early on. But what we have is very promising because the animals are free to behave. They move around their tank, they do whatever this, when they have this thing, bizarrely enough, they can't, they don't, I don't know if it's bizarrely enough. I wouldn't say bizarrely enough, but they don't really feel, the logger unit, which is in the mantle. It doesn't bother them. They don't investigate it all the time. They have one incision, so the entire implant and implanting the logger and everything happens through a single 2 centimeter incision. They will sort of touch that because it was a wound that was closed, but that's it. That's their only reaction to the entire procedure. And so, in that condition, the idea of being able to work with them during learning is very hopeful, because to get an animal to behave normally, they have to feel okay. A lot of experiments work on negative reinforcement, but negative reinforcement is, it's a different circuit. We work with positive reinforcement learning, which is what I'm also interested in.
And how do they perform under those circumstances? Like, what's their rate of learning in most tasks?
It's a very, very difficult topic, because when you're dealing with animals that are not the standard lab animals, you realize there are very big differences in what performance is. Octopuses, we pretty much manage to do about 10 trials every other day reliably. They won't eat food that isn't too small. They won't eat small pieces of food. And so you have to figure out what their favorite tasty food is. But what you can't do with octopuses is what you do a lot in other animals, which is food deprivation or water deprivation or some sort of thing where they're not suffering deprived, but that increases their motivation. And if you try to do that with an octopus, you're going to get an animal that is just unresponsive to the point that all it's going to do is attack whatever you do. So basically, you have an animal that's going to do about 10 trials every other day, and their success rate is never going to be the same as 100 tests in a pigeon per day. 100 pecks or 100 nose pokes of a mouse, which can be very frustrating, but they learn. They do learn. It depends on how difficult the task is. A lot of the tasks that I did were difficult because they require the animals to use and gather information just from one arm. And they learn to put their arm into a specific thing in the maze and then just learn from information from that arm. And that takes a long time. Just learning a simple visual association is very fast. And by very fast, I don't mean mouse fast or rat fast, for that matter, or turtle fast, for that matter.
Well, you could conduct some learning experiments while also taking these voltage measurements, but I don't know how you go from one to the other. Is that too big of a leap to take all at once, or how do you look at connecting your raw data to some of the more higher level cognitive activities?
Well, I mean, I think at the moment, so when I talked about this cellular form of learning, this long-term potentiation, which means that, when an input neuron goes into an output neuron, you get a certain reaction from that second neuron. And then the cellular form of learning is basically because you have several input signals, then that second neuron is going to react differently, either higher reaction or lower reaction, and that was shown in the area in which we did our initial recordings. And that is an area that, from lesion experiments, they've suggested has to do with visual learning. And visual learning, when it's a simple association, is fairly fast with these guys. We're also looking at ways to be able to start and stop the loggers so that, in a one chunk recording, it would be very hard to do the entire procedure.
And I'm curious, do you see any opportunity for artificial intelligence to play a role in your work in the future?
I think I don't know enough about it to see how it would contribute. I think and the artificial intelligence is probably more intelligent than me on the subject. Maybe. I don't know.
If you don't mind my thoughts, I think actually some of the boring stuff it might be able to help with. You have all this video, and I see in the paper like you have it labeled, they're sleeping or there's this activity.
Yeah.
And that's probably a difficult job for a few grad students or maybe yourself, and a lot of tagging. That could probably be done by AI.
Yeah. So that is being done in several places that people have started to automate that. Again, the technology is now there, whereas before it was pretty much impossible to automated track an octopus that has no fixed shape. I mean, usually you just got the computer to say, recognize this shape, now track that. And if there is no fixed shape, then good luck. And now there are places that are doing that. And they're actually looking and also modeling arm motion and things like that to try and figure out what are the different influences, for instance, for an arm to move in a specific way or to follow some specific contour or things like that. So, I mean, this stuff is being done now. And it's very cool what's happening.
And then I guess where does your research directly go next? What sorts of things are on the horizon for you?
I think, I mean, at the moment, I continue focusing on hopefully having a chance to improve on the neural recordings and also look at sensory motor control in the arms from a behavioral approach, because I have a lot of patience, apparently.
Well, I also wanted to touch base on some of your work with tortoises. I guess to kick it off, how did you get from octopi to tortoises?
That's a personal story rather than a scientific story.
All right.
My now husband, who I met working on octopuses, had moved to Vienna, was working with the Vienna Zoo, and he gave me a call and he said, "Wow, you have to see these turtles. They're amazing." And I said, "Wow, that sounds so boring." and then I visited and I fell in love. And
Sorry, with your husband or with the tortoises?
With the tortoises. No. No, and I met them and they were amazing, and that was 9 years project, including 7 years long-term memory. It was really working together with zoos. You get to do a lot of things that are not possible in other settings, because nobody can keep animals like that. There is no lab that can keep 100-year tortoises. Our oldest subject, who sadly died a few years ago, was over 100 years old, George. And he had been in the zoo since the 1950s, I think. So that's not, I mean, I also worked with stingrays, another animal that you can't really work with anywhere else. And that aspect of my work is more trying to find, it's basically finding intelligence in animals because we never had the chance to test them in a way. So I mean, with the stingrays, well, they're sharks, and then a lot of people just consider it like a nose, an animal and a nose that swims around. And with turtles, it's like, okay, giant rocks. And exactly the response that I had, which is, wow, that sounds extremely boring. Yeah, it's just because we never had the chance to look. And when you do, it's quite fascinating because for each one of these species, you have to figure out a different way to ask the same question. I'm not going to have a turtle stick its arm into a maze. That's not going to work. If I try, my result will be that, well, the turtle is not very smart. Actually, if I take the turtle and I try to teach it a simple operant task, they'll learn it in one session. It's a question, so it's a lot of figuring out how you can ask a similar question to completely different animals with completely different ways of interacting with the world.
Yeah, I was quite surprised by some of the things I learned in your paper on tortoises having to do with their memory. Could you just give us the experimental setup and some of the results?
So when we started, we did a very simple operant training, starting with a target ball. Okay, this is a boomer ball for a dog, but now this is what's going to get you a carrot. If you walk up to this and bite it, and only it, not the stick, not this, not looking behind my back, not any of the other ways you were trying to get that carrot, only if you bite this ball, you're going to get a carrot. And that was the first basic task, and the learning speed for that was, I mean, for some animals it was within that session that we started, and other animals take longer. Then we did color discrimination with them, and so we assigned each animal a different color ball. And when we did this, we actually did it in two different settings. We did it in two different zoos. So in one zoo, we did it with all of the animals being in the same enclosure at the same time. The animals could, if they chose to, watch another animal interact with us. We didn't make them watch. We did stop them from interfering, so we didn't let them get in the way, but they could, if they wanted to, look. And in the Vienna Zoo, we separated each animal to work with them. So each animal, they pretty much lined up. They knew it was time to work, and I opened the gate, the turtle goes out, and the other one is already walking in for his turn. And so our first result was actually to see that the observation of another turtle learning and doing this task resulted in a faster learning speed. And this was really from the observation, because when we did the color discrimination and suddenly every turtle had their own color ball, well, that was the same. Watching someone else learn that didn't help you because he had a different color ball than you. Then after we did these experiments, we came back to the zoo in Vienna after several months, I think. The first time we did it after several months, and we redid the color discrimination experiment, and the learning was faster. So it wasn't immediately that they recalled, oh, okay, my ball is green, but they relearn it much faster. And they remember the basic task being ball, bite ball, get, well, carrot or beetroot, depends on the type of turtle. Then 7 years later, I think it was 7 years later, we came back. At that time, there were only 3 turtles remaining in the Vienna Zoo. The other ones had to be given away because they were teenagers and getting very bitey. And Galapagos turtles eventually will become larger than Aldabra. And the Aldabra had been there since, well, George had been there since the 1950s. So the Galapagos were rehomed to other zoos. So we had the 3 Aldabra tortoises remaining, and we retested the basic, well, here we are walking in with this target on a stick. which they immediately remember. They immediately remember why we're there and what they're supposed to do. One of the things that I like mentioning when talking about this is, again, that we never do any sort of food deprivation or this. We just removed carrots. When we did the initial training, we removed the orange stuff from their regular food and gave it to them as the treats. But the truth is, they didn't do it for the treats. They were just interested in the interaction. They lined up for. And sometimes, unfortunately, the carrot managed to get into their food, but that didn't still stop them from wanting to interact anyway, wanting to play. I don't know. I call it play, but it's not play. They're wanting to interact.
Too bad the octopi don't like carrots.
No, they don't. They like shrimp. They like pieces of shrimp, clams. With the octopuses, you do something else. In the octopuses, what we do is we basically replace feeding, where usually you just give one piece of large food every other day. And we replace that with feeding through the training, through the experiment. So we always make sure they get the right amount of food. It's just that they get it in 10 pieces rather than as one chunk.
I wanted to ask if you had any advice for young aspiring researchers looking to get into this field and specific areas of research you work in.
I think for behavior aspects, I always think that trying to not look at it from our point of view is important. I think in general, also science, it's just creativity. It's trying to find its creativity. But I don't know. I don't have specific advice.
That's all right. As long as it's not do something else.
There is that. There is that. There is don't expect to make any money.
But it sounds like you could expect to advance the science pretty quickly, maybe.
Yeah, there are a lot of new labs who are putting the newest techniques into cephalopod research, and that's very exciting. And there are a lot of places that are doing that. So it's a small field, but it's pretty exciting, and it's definitely fascinating, and it has no real practical applications. That's, I think, something that is very important to know.
For now.
Yeah. No, knowledge is knowledge.
Yeah, and they found...
And understanding is understanding.
And they found CRISPR on accident, from what I understand.
A lot of things have been discovered by accident. And basic science definitely should continue to have a very important role in science.
Absolutely. Well, Tamar, thank you so much for taking the time to come on and share your work.
I hope I at least made one serious answer.
Oh, absolutely. Yeah.